ARP23 is a protein complex that nucleates branching microfilaments from existing microfilaments. Its ability to nucleate microfilaments is primarily due to its subunits ARP2 and ARP3, and resembling the structure of the G-actin monomers found at the growing plus end of an actin filament. Focusing on the effect of pH on protein structure, changes in pH levels are associated with changes in hydrogen ion concentration. Different ion charges are found in the side chains of amino acids, which influences the folding of globular structures. For instance, nonpolar side chains form the hydrophobic core, and polar charged side chains form the hydrophilic exterior of globular proteins. As a result, changes in hydrogen ion concentration can lead to altered protein structures and subsequently altered protein functions. This research proposal seeks to determine the specific functional impairment of the ARP23 complex as a result of its cytoplasmic environment undergoing a change in pH. This led us to forming the research question: what is the effect of the pH of the host cell's intracellular environment on the regulation of actin polymerization by the ARP23 complex? Based on this research question, it is hypothesized that the pH of the host cell's intracellular environment has an effect on the regulation of actin polymerization by the ARP23 complex. Prediction 1 is that increasing the pH of the host cell's intracellular environment impairs the ability of the ARP23 complex to nucleate branching microfilaments. So method 1 focuses on using the FRAP technique, fluorescence recovery after photobleaching. So for the experimental group, the intracellular pH of mouse embryonic fibroblasts is altered. To achieve this, these cells are rinsed with 3 milliliters of DMEB, Dolbeco's modified essential medium without bicarbonate, supplemented with 20 millimoles of Heap's buffer solution, 4.5 grams per liters of glucose, and 10% FCS, fetal calf serum, which has a 7.4 pH, DMEB per FCS. The fibroblasts are incubated at 37 degrees Celsius for 30 minutes in 3 milliliters of the same medium containing 20 millimoles of NH4Cl. These steps are repeated 2 more times except using a DMEB FCS pH of 4.4 and 1.4. The intracellular pH measurements were determined based on amounts of C14 labeled benzoic acid in the cytoplasm. The control group consists of mouse embryonic fibroblast conditioned media untreated with intracellular pH altering media. Next, actin filaments are labeled with fluorescein phalloidin, which is a toxin which binds to F-actin with high affinity and is conjugated to the green fluorescent dye fluorescein, FITC. ARP23 is labeled using an antibody to the P34 subunit of the protein complex and CY3 red fluorescent dye. Using a micrograph, the fluorescently labeled actin filaments and ARP23 complexes in the lamellipodia of the fibroblasts are viewed. The amount of daughter filament microfilament branches associated with the ARP23 complex were recorded for each intracellular pH level. In the experimental group, lowering the intracellular pH should impair the formation of branched microfilaments nucleated by ARP23. Prediction 2 is that increasing the pH of the host cell's intracellular environment impairs the ability of the ARP23 complex to cap the minus end of the daughter filaments. Method 2 focuses on using the FRAP technique. Intracellular pH approximately 7.4, 4.4, and 1.4. For the control group, maintain a constant intracellular pH by not treating the MEF conditioned medium with intracellular pH altering medium. In addition, fluorescently label the actin filaments and ARP23 complexes using the same labeling agents that were used in method 1. Using a micrograph, view these fluorescently labeled structures at the lamellipodia of the cell, where microfilament branching networks are expected to occur. Record the number of G-actin monomers depolymerizing from the minus end of the branching microfilaments associated with the ARP23 complexes for each intracellular pH level. In the experimental group, lowering the intracellular pH should impair the ability of the ARP2/3 complex to inhibit depolymerization at the minus end.